In this video, we're going to go over prokaryotic RNA transcription. And we're just going to start with the basics. And the enzyme that carries out transcription for us are called RNA polymerases or RNAPs. And what they'll do is they'll read in DNA and create RNA. Now these are different than the DNA polymerases that we talked about in that we don't need a primer. We can just take two RNAs and connect them de novo. Eukaryotic cells can have 4 to 5 different RNAPs, and each one of these is going to make a different class of RNA. So mRNA, tRNA, rRNA. Now let's take a look at the structure of an RNAP, the E. coli one to be specific, and these are massive proteins. So the E. coli RNAP's 449 kilodaltons made up of a bunch of different subunits. And they're so big that we can see them through an electron microscope. Most proteins you can't see through an electron microscope, but these things show up nice and big. Now, the way to look at this is that the RNAP looks like a claw with our beta and our beta prime forming the pincer unit, and that's in your cyan and your pink colors. Right now, we are looking at the open conformation, which is occupied by double-stranded DNA. That DNA is shown in blue and green. Now what happens is that your template DNA, which is shown here in green, will continue to the active site at the end of the main channel. And it's going to be base pairing with incoming nucleotides at a site called I+1, which has magnesiums near it. And this shouldn't be too much of a shock, as this is a polymerase, and we saw in the DNA polymerases we also had magnesium. Now the RNA, which is shown here in red, the 3' N will form a helix with the 5' N of the DNA, and you're going to be exiting through a channel between the beta and the beta prime that we call the RNA exit channel. Now, when this polymerase binds a specific part of the DNA called a promoter, and a promoter is a sequence that specifies where transcription should start, the subunit sigma will extend across the core enzyme. And when it does this, the pincers actually close and become shut. This is called the closed complex. When this happens, the sigma subunit will open the transcription bubble by binding the non-template strand -10 promoter element. Remember, these numbers mean how far away are you from initiation, where you start encoding the gene. So -10 element would be -10 positions away from that. Now, most of our protein is negatively charged. However, the amino acids that interact with our nucleotides are positively charged. And that makes sense. All this negative charge is kind of directing the nucleotides to interact with the bases that we want that are positively charged. Now, we need to understand some terminology when we're talking about transcription. And so, for the DNA itself, we have two different sides, the sense or coding strand and the antisense or non-coding strand. So when we get to the initiator, the sigma is going to create the transcription bubble, right? And the sense strand, which is coding, that's what the RNA is gonna have the same nucleotides as. So, for example, if our sense strand was ATG, the RNA would be AUG. So our other side, our anti-sense strand, is called our non-coding strand. But this serves as our template. This is what we'll be reading to make the sense strand, or the RNA rather. Now, since we're talking about prokaryotes, we have to understand how genes are ordered in prokaryotes. So for eukaryotes, when we have a protein coding gene, we usually read those individually. That is one read for one gene. But for prokaryotes, it's different. You often have multiple genes just lined up right next to each other. And these units are called operons. And the genes that are lined up usually have related functions. It's just not random. These genes are usually go together. So for our example of this operon, we're going to look at the lac operon. And we have three genes that are involved in lactose metabolism. And there also contains sequences that control their transcription. Now, the operon, like I said, is a single unit, so you get a long mRNA from this. While eukaryotes, you don't. There's just one gene. So a prokaryote is called polycistronic mRNA, and a eukaryote is monocistronic mRNA. Now let's look at the promoter sequences that are in the sense strand. So the RNAPs will bind at these initiation sites that are promoters, and certain sigma factors will recognize certain promoters better. So our non-template strand, our sense strand, that's where we're gonna get our actual sequence from. And if we look at the numbers here, I mentioned this in our previous slide where we're looking at these numbers. Initiation, we call that +1. Anything downstream, we get a negative number. Anything upstream, we have a positive number. And note, there is no zero base, so the immediately to the left of +1 would be -1. And what we can see here is that you have certain consensus regions. That is, if we look at different genes and just line them up, we see, at the -10 region, we have something that we have called the Prinball box. That is, we always see more or less the same nucleotides there. Same thing at the -35 region. In general, we see the same types of nucleotides there, no matter what gene we're looking at. And so here we have a consensus sequence which is saying like 69% of the time we expect a T, 79 a T here, so on and so forth. Interestingly, on the initiation site, it's more or less scattered. So I know these don't add up to 100, but this is basically showing you the higher the number, the more likely it is to be found there. And so here you're going to find just about any nucleotide to start at the initiation site. Now, the way that these sequences were determined is through footprinting. So what you do for footprinting is that you have your protein bind to your DNA, and then you remove all the DNA through alkylation. So when you do that, you can see which DNA was bound to your protein. So that's how the Prinball box in the -35 region were discovered. Now, at the initiation site, I said that you're going to find like all amino acids there more or less equally. Well, at the +1 site, we do have some specificity, so that's going to be A and G. And so that'll be flanked between a C and a T for the most part. You can see that 55, 48, so only roughly half the time there. But for the most part, at the +1 site, you're going to have that A or you're going to have the G. Now, if you have mutations in these conserved regions, that might greatly increase or decrease your promoter's initiation efficiency. It really depends on the mutation. And what's interesting is that if you want genes to be transcribed faster, you can just change your promoter, because what really determines the speed is how well your initiation complex and how well your DNA can interact with the RNAP holoenzyme. So for example, let's say you have a mutation in the -10 region. Now you have a much higher affinity for the RNAP holoenzyme, which can make the initiation complex. That gene is going to be transcribed a lot more than before the mutation. And that's it for this video. I will see you in the next video.